Hi everyone, welcome to this talk on simulating single-cell RNA-seq data controlled by causal gene regulatory network. My name is Yazdan and I'm a student here at McGill in Ahmad's lab. And let's start today's talk with a quick background on gene regulatory networks, or GRNs. As you know, gene expression is controlled by interactions of genes and their various regulatory elements. And we can think of these interactions as a graph and also model GRNs as such. And this data structure now allows us to use existing computational methods and algorithms to simplify our studies. And very often in our field of research, GRNs are simplified into a bipartite graph between transcription factors and target genes. So we have a set of nodes that are transcription factors, or TFs, and a second set of nodes comprised only of target genes. And regulatory interactions are represented by edges outgoing from a TF into target genes that TF regulates. And although it's an incomplete model, we still get a lot of insight into gene regulation, which helps us model and also understand various biological processes. And because of that, there has been quite a large interest in inferring GRNs from single-cell data. And with that, a need to benchmark GRN inference methods in a systematic way. And to do so, we need two things. One of them is a single-cell dataset, and the second one is a corresponding ground truth GRN. And this is where we face a challenge, because how do we even come up with a ground truth GRN? Well, we could construct that from known interactions from literature. It's very hard to systematically do so, but even if we do, we might end up with GRNs that have missing edges. Or we can rely on structured biological databases such as KEGG and I2D, or on ChIP-seq datasets. But the common issue with all these ways of creating ground truth is that they give us zero guarantees on whether it corresponds to the system we are studying, because gene regulation is very context specific and also cell type specific. So to ensure that the created ground truth are true to the system under study, we really need to perform perturbation experiments on that same system, or on a system that closely mimics the conditions used for GRN inference. And those experiments are very long and costly. So the next logical step would be to use synthetic data for benchmarking. And we also face challenges in this area because there's a gap in the ability of simulators to closely mimic real data. And there have been studies such as Beeline where they found that the best performing methods on synthetic data were the worst performing ones when tasked to infer GRN on real experimental data sets. And that gap is very huge. And simply, most simulators simply do not incorporate the GRN, and the very few that do, such as GeneNet Weaver, many of them correspond to the bulk RNA-seq domain, and those cannot be directly applied to single-cell datasets. And there are very few tools and simulators for single-cell. One of them is Sergio, and most of them require extensive user manipulation. For example, Sergio requires a user to statistically match its output datasets to a real reference dataset by, in an iterative fashion, manually adding technical noise to its created datasets. And there is no guarantee that after that, all that technical noise has been added, that the causal TF gene relation remains in the dataset. So we propose a new method called GroundGAN, and GroundGAN is a causal implicit generative model, which as input accepts two things. One of them is a single-cell expression matrix. The second one is a user-provided GRN, and it's trained in two steps. So the first training step consists of training a Wasserstein GAN and all TFs and genes, and once this GAN is trained, we isolate the generator and freeze it and rename it to a causal controller, which now only generates TFs. Now these generated TFs, and along with noise, are passed into a second GAN, which is again a Wasserstein GAN, and the generator of this GAN is called a target generator, and this is where we impose the user-provided GRN by making the target generator's neural connection follow the connections in the GRN, and this ensures that every single target gene is generated as under the control of its regulating transcription factors. And we train the second GAN as any other Wasserstein GAN, with the addition that we have auxiliary networks which further impose the GRN and ensure that the GAN is causal. But how does GroundGAN perform? Well, we ran it on two different data sets. One of them is a human PBMC data set, which is highly heterogeneous, has many different cell types, and it's quite large in terms of number of samples. And the other one is a bone marrow data set, which is quite the opposite. It's not a steady state data set, it's a differentiation data set of progenitors into different fates and has very few number of samples. But in both data sets, we can see that real and generated cells, which are shown in blue and red respectively, they overlap in this 2D space. And in the case of human PBMC data set, we can see that GroundGAN is able to generate from all different cell types, even the cell types that have less samples, which are now isolated in these 3 little islands you can see. So GroundGAN does not face the common issue of mode collapse, where the generative model learns to only generate parts of the data set. In addition to that, we have different quantitative measures of measuring how realistic GroundGAN data really is. So when a random forest is trained to distinguish between real and GroundGAN data, it performs quite close to random. So GroundGAN successfully fools this RF, and the RF ends up with an AUROC of 0.54. And in terms of different distance metrics such as cosine, Euclidean distance, and Jensen-Shannon divergence, and other scores such as median integration literacy and maximum mean discrepancy, GroundGAN still outperforms state-of-the-art methods on most of them. And although it is constrained by a GRN that is imposed onto the model, but we believe that GRN is a way of integrating domain knowledge into simulation, which helps us get better results in terms of mimicking realistic data. But that is not enough for GroundGAN, because we also wanted to show that it causally enforces the GRN. So in this experiment, we inferred a GRN on real data using GRN Boost 2, and for each gene we impose half of its interactions onto GroundGAN as positive control and generated data, and the other half we did not impose, which now creates our negative control. And we can see that the edges that were imposed as part of the positive control are now picked up by GRN inference methods, and those edges are assigned higher scores than their scores in real data. So their AUPRC in this case goes up from 0.29 to 0.77, and edges in the negative control that were not imposed are now assigned close to random scores by GRN inference methods, and their importance goes down from 0.24 in real to 0.08. So GroundGAN not only enforces the edges that were imposed, but also messes up the causal relation of the edges that were not imposed through it. And on the same subject, we have a second experiment where we perturb TFs one by one, and we verify that the genes not regulated by TF remain unchanged in terms of expression. And to quantify how much the genes that were regulated by the perturbed TF change, we perform a two-sided Wilcoxon sign rank test, and we obtain mainly very small p-values under 0.05. And we talked how GroundGAN was a causal implicit generative model, and that opens many doors. One of them is that we can sample from both interventional distributions and observational distributions. And sampling from interventional distributions allows us to generate samples that do not naturally occur in the dataset. And one example is the ability of performing in silico knockout experiments. So in here, for example, in the middle figure, we knocked out the top 3 differentially expressed TFs of the CD56 plus NK cell type. In here it's in cyan. And GroundGAN does not generate cells for that cell type anymore. And the cells that used to be there are now dispersed into its neighboring cell types. And coming back to our original goal, which was benchmarking GRN inference methods, we used GroundGAN's imposed GRN as ground truth, so that is how we tackle the issue of lack of gold standard ground truth when it comes to benchmarking. And we then use the beeline pipeline to benchmark different GRN inference methods, many that required pseudotime information. And what we found was that PIDC, GNE3, and GRN Boost 2 were the best performing ones on both datasets. And we talked previously about how many methods, the ones that required pseudotime, perform really well when ran on synthetic data, but their performance dropped close to random when real data was used. And our results concord with Beeline's results on real experimental data sets and curated models, and not their results on simulated data. So GroundGAN also closes the existing gap between benchmarks on simulated and real data sets. So in conclusion, we established that there is a lack of gold standard ground truth when it comes to benchmarking GRN inference methods, especially on single-cell data. And to that we proposed GroundGAN, which is a simulator that generates realistic single-cell data sets, and while doing so preserves gene identities and pseudotime ordering of cells. And preserving pseudotime ordering allows us to benchmark GRN inference methods that require pseudotime ordering. And while it generates genes in a way where they're causally expressed under the control of the regulating TFs, and captures technical and biological noise with no user manipulation, and only implicit parameterization. And because GroundGAN is a causal implicit generative model, it can be used to perform in silico perturbation experiments and knockout experiments. And very importantly, bridges the gap between synthetic data and real data benchmarks. So I want to very quickly thank the members of my lab for their feedback and support. And if you have any questions, I'll be happy to answer them. Just drop them down in the question section. And thank you very much for attending this talk.